So in this lesson, we're going to talk about DNA replication, and it's fairly simple, but one of the most important things is that you do want to be able to understand where this is taking place. So we're going to restrict our discussion to DNA replication in eukaryotic cells, and please remember that eukaryotic means that there's a membrane-bound nucleus and the DNA is housed in that membrane-bound nucleus. So we know that DNA is a double-stranded molecule, and DNA replication actually occurs in the nucleus, okay, not in the cytoplasm, not in another organelle. DNA replication, as the cell prepares for cell division, happens during interphase of the cell cycle, and it is always happening inside the nucleus of a eukaryotic cell. So when we take a look at how DNA replication actually occurs and what the end result is an exact copy of the DNA. However, it's a semi-conservative process. So that means that because we're replicating the DNA strand, we end up with a new molecule of DNA that is half from the original and half new. So if we look at that visually, we can take a look at the graphic at the bottom here, which shows that we start with an original DNA strand, and then at the end of a DNA replication, we have two DNA strands that are identical to each other, okay? And you notice that half of it is blue and half of it is purple. So that means that the purple part is actually conserved from the original strand. So each of the resulting DNA strands has a purple section, which is conserved from the original part. But then we also have this blue section, which is newly formed. And so that's why it's called a semi-conservative model, because part of it is conserved from the original DNA strand and part is newly formed. Now, when we talk about the actual process of DNA replication, it actually happens in 3 stages. So stage 1 is that we have to unwind and separate the DNA strands. So remember, the DNA is in that characteristic double helix structure. So prior to DNA replication, the DNA strand is going to unwind, and so that it's more in that ladder formation. And then we actually are going to have to break those hydrogen bonds that hold the complementary base pairs together. After we have the separation of the DNA strand, then we are going to build complementary strands to that. So we're going to work off that old DNA strand and create a new part. And then lastly, we're going to have certain enzymes that actually check for errors and correct any errors that there might be before the final strand is all kind of glued back together and ready to go in a new cell. So if we take a look at our graphic here, we can see the generals of when we discuss DNA replication, and we kind of go in terms of number here. Now, the other thing I want you to pay attention to here is you'll notice there's this arrow down at the bottom, and it says that this is the overall direction of replication. So when you think of this as kind of being the DNA molecule being unzipped, I want you to notice that we are always kind of moving into the part that still needs to be unzipped, and we call that the replication fork. So we are always moving into the replication fork and unzipping that molecule. So always kind of look for that overall direction of replication arrow. If you don't see that, just the ends that are already split apart over here, that's going to be already completed, because you can see we already have an old and a new strand being formed. So you always go into that replication fork. So the first thing that happens is helicase, which is a very special enzyme, and you can think of that enzyme being the unzipper of the DNA molecule. So what the helicase does is it forces itself into that replication fork, and it actually breaks the hydrogen bonds that are holding the base pairs together, okay? The next thing that'll happen is, remember that those base pairs are really attracted to one another. So the A and the T always wants to go back together, the C and the G always wants to go back together, and so we actually have these special proteins called SSBPs, or single strand binding proteins, that actually stabilize that unwound DNA and prevent those bonds from re-engaging with one another, because then you would just basically be unzipping and then zipping and then unzipping and then zipping. So they help prevent the re-engagement of those bonds between those base pairs. The next thing that'll happen is we have a special enzyme called DNA polymerase, and DNA polymerase I want you to think of as the builder of the DNA strand. So what DNA polymerase does is it actually brings the appropriate base pair to complement the base pair that is on the original DNA strand. So for example, if the original DNA strand in this first base pair has an A, then the DNA polymerase rather is going to bring a T. If the second one is a G, then the DNA polymerase is going to bring a C, and brings those base pairs to actually build that light blue new DNA strand. The other thing you'll notice is it always works and builds in the 5' to 3' direction. What that means is that the 5' to 3' is the direction in which the base pairs are brought to the new DNA strand by that DNA polymerase. And the one that is built first we call the leading strand. So leading, that means you're first, right? So that's the leading strand. So it's built continuously, and it's built quickly, and it's built in the 5' to 3' direction. Now you'll notice down here at the bottom that we actually have a different scenario going on here, and you can see that we have these little kind of breaks in the light blue newly formed DNA strand. So this is called the lagging strand, and it lags behind because it is not built continuously. And the reason it's not built continuously is because it's being built in the opposite direction. So instead of being built into the replication fork, it's actually being built this way, and therefore it is slower. All right, so these chunks of DNA that are not fused together, okay, see how we have all these pieces of DNA here that are not yet fused together? We call those Okazaki fragments, named after the scientists that actually discovered them during the process of studying DNA replication. And so you'll notice that the leading strand back at the top does not have those Okazaki fragments. The lagging strand, because it's being built in the opposite direction, going away from the replication fork, actually has those Okazaki fragments. Now, the last thing that happens is once the DNA polymerase has brought all of those base pairs to both the leading and the lagging strand, then the DNA polymerase will actually go back and it'll proofread and it'll check to make sure that those base pairs were added properly, so that we don't have a C where a T should be, or an A where a G should be, and it kind of does like the proofreading really of that. And then the last thing that happens is ligase, which is another enzyme, so we can see that one here. What DNA ligase does is it actually glues, so you can think of that as the glue. It actually glues together those Okazaki fragments and ensures the integrity of that DNA strand so that they're all fused together properly to the growing strand. Now you'll notice there's another thing on here called the RNA primer, and the RNA primer is really just a little piece, so that's that kind of dark, kind of purpley color thing here, or dark red thing on the lagging strand. So notice that you have the RNA primer on the lagging strand only, and that is to show the DNA polymerase where to start and where to work. Okay, so really that's what the RNA primer is for. It's a little teeny tiny piece of RNA that is actually for to direct the DNA polymerase to the correct place in terms of building that strand of DNA on the lagging strand. Now, if we take a look at just a little more detail based off of the diagram that we just previously looked at, please remember that helicase is the enzyme that first engages with DNA in the nucleus prior to DNA replication. So I want you to think of DNA as the unzipper of the DNA molecule. So this is going to disrupt and break those hydrogen bonds that are holding those base pairs together. Next, we have those SSBPs, we talked about that previously, and those are those single-stranded binding proteins, and what those do is they actually bind to the separated strands to prevent those base pairs from re-engaging and those hydrogen bonds from reforming, because if there weren't those SSBPs, then literally the helicase would break the bonds and then they would go and reform, and helicase would break the bonds and then they go and reform, which is obviously counterproductive when you're trying to keep that strand separate for replication. Next, we have the place where the helicase is kind of working into and separating into, and the helicase is going to work always into the replication fork to actually separate those hydrogen bonds and break those apart, okay? And when we talk about replication, please remember that the helicase always works moving into that replication fork and spreading the sides of the DNA molecule apart. RNA primase is an enzyme that will actually make a primer, and a primer, just think about what a primer is. A primer is like something that starts things, right? So RNA primase creates this little piece of RNA called a primer, and what that primer does is it actually attaches itself at certain places to show the DNA helicase where it actually needs to go and where it actually needs to work, okay? So RNA primase is an enzyme that produces a little pieces of RNA called RNA primers, and the RNA primer basically guides the DNA polymerase to the correct place to actually form the newly formed DNA strand. When we actually build the complementary base pair strands, remember that we are always talking about DNA polymerase here. So DNA polymerase is the builder. It's going to pluck off those RNA primers and it's going to replace them with the appropriate base pair. So if the original parent strand had an A, the DNA polymerase is going to bring a T. If the original parent DNA strand or template strand had a G, the DNA polymerase is going to bring a C. So DNA polymerase is essential for actually building the new strands on both the leading and the lagging DNA strands. The DNA polymerase is also very important for proofreading and making sure that all of the base pairs were allocated appropriately based on what the DNA template was asking for. So if there's a T, there's an A. If there's a C, there's a G. If there's a mistake, then the DNA polymerase will literally remove the base pair that was added incorrectly and put the correct base pair there instead. DNA polymerase always works in the 5' to 3' direction, and that's why we end up having the leading strand, which is completed in a continuous strand, and then we have the lagging strand, which has the Okazaki fragments, because it's actually working away from the replication fork, and as a result, it is done much more slowly and cannot be completed continuously, because it literally has to kind of go backwards away from that replication fork. And so then we end up with those little Okazaki fragments that later have to be glued together by the ligase. And so that's our last enzyme, is the ligase. So think of it as glue. And what the ligase does is it actually links and glues those nucleotides together for the final formation and completion of the new DNA strand. When DNA repair is happening, again, as we already said on the previous slide, there's more than one type of DNA polymerase, but we just kind of talk about it generally speaking. So DNA polymerase is the builder, bringing those base pairs, and it's also the proofreader. So remember, it's also going to check to make sure there's any mismatches in those base pairs, and if there is, the DNA polymerase is going to fix that. And then when mismatches do happen, the DNA polymerase will remove the incorrect nucleotide, replace it with an appropriate one, and then the DNA ligase is going to glue that nucleotide into place. Because remember, even though I say like, oh, it's going to bring a base pair, remember the nucleotide is the monomer of the DNA, right? So it all moves together. So that phosphate group, the deoxyribose sugar, and the nitrogenous base are going to all move together, okay? It's not like you just have these random little base pairs floating around. The monomer, the entire nucleotide moves together and is pulled all together in its entirety by the DNA